So just a little bit of background on Zika virus. So there were like 4 million outbreaks in the world. There were 5,000 cases in the United States. It's like passed from a woman to her pregnant child, or a pregnant woman to her child during birth, and we all know that causes microcephalies, which is an abnormal brain like development, and that's not good. It's like small heads. And also, one of the main problems with the Zika virus is that there actually is no vaccine, so that's why we need antiviral drugs like these. And why do you care? So I'm here and talking to you about this, but why should you care? You should care because people were afraid of Zika virus, people were afraid to travel, people were afraid to have babies, people were afraid to get pregnant, and that's just not okay. That's a problem. And with a possibility of another outbreak of Zika virus, we should always just have a potential solution, and we now do have that with this new antiviral drug. So with that said, the name of my paper was the clinically approved antiviral drug sofosbuvir inhibits Zika virus replication. So just some background information on the drug that I'm presenting on. It's called sofosbuvir, but it was actually approved in 2013 for treating hepatitis C. But because they thought hepatitis C and Zika virus were like pretty close viruses, they're in the same family called the Flaviviridae family, they decided that maybe if it works against hepatitis C, it can also work against Zika virus. And that's what they decided. And you can see that they're actually very structurally similar. The top one is the Zika virus, and then the bottom one is the hepatitis C. So I mentioned this before, but there is no vaccine, but this is an antiviral drug. So the difference between those two is that a vaccine prevents the infection because you're given a little bit of the infection before it, and then it induces a positive response for your body. And an antiviral drug, it fights the infection after the virus has already entered your system, and to do that, it has to be given after, and then it prevents it from replicating. And this we're talking about today is an antiviral drug. So now we're getting to the interesting part. How does sofosbuvir actually interact with Zika virus? So first, it targets the RNA polymerase, and this is the amino acid sequence that it needs to bind with the RNA polymerase because it can't just do that without anything. So we talked about RNA polymerization, and that's important because the RNA polymerase copies the virus's RNA so it can replicate, and if we stop that function, then it no longer can replicate, and if it can't replicate, then there's going to be less Zika in the cell, or in the body, and that's good. And here, this is exactly how it does replicate. The sofosbuvir actually takes like the structure of this, which is a nucleotide, and nucleotides are added every time the RNA polymerase tries to copy a part of the RNA, and usually after a certain number of nucleotides, it'll try to add this nucleotide called the UTP nucleotide, but instead, because these two are so structurally similar, it'll add the sofosbuvir instead. It'll add the sofosbuvir instead, and when it does that, no other nucleotides can bind to this phosphate group right here because it's actually different from the actual triphosphate group that should be there. And if that can't bind to it, then no more nucleotides can be added, and then the RNA polymerase stops replicating the RNA. So this is just the mechanism of it, like simpler. So you can see that this is different from that, but these two parts are exactly the same, which is why the RNA polymerase sometimes mistakes the UTP nucleotide for the sofosbuvir. And this residue right here prevents the RNA polymerase from adding more nucleotides. Is that clear? Does anyone have any questions? Awesome. Oh, yeah.
So this targets specifically the RNA polymerase that's coded by the virus, or is this not one that would be in the host cell?
I think it's the one in the host cell, but it like targets the one in the host cell, so when the virus is already in the host cell, it can't replicate more viruses.
So this potentially would stop all transcription?
Yes. I think so, but I'm not entirely sure. So this is like the next, this is also part of the mechanism, just to make it like a little more clear. So here's nucleotide A, and this is what should happen. It should add a nucleotide and then another nucleotide and just keep going. As the RNA polymerase moves, it'll keep adding nucleotides. And then down here, this is what happens when the soft phosphobutyric is in the system. So it'll add the nucleotide, and then it'll mistaken this for the nucleotide B. So it thinks it's adding the right nucleotide, but it's actually adding this antiviral drug, and that prevents the RNA polymerase from adding more nucleotides. So they did some studies. This was one of the studies, and basically what happens in this graph is that it measures relative fluorescence units, and those measure how much RNA polymerization is actually happening. So you can see that as the drug concentration goes up, STP represents the soft phosphobutyric, and that actually goes down. So we can see that there's less RNA polymerization happening, and if there's less RNA polymerization happening, that's good because then there's less replication happening. And then these are just this is the negative control, this is the positive control, and it obviously just matches up with the positive control. And that drug works, but we don't use that drug because it's harmful to pregnant women, and that's kind of what Zika virus is a lot of it is about. How does soft phosphobutyric actually affect individual cells? So you can see that with soft phosphobutyric, Zika production in most of the cells goes down really quickly, but with these two cells, it actually doesn't. It just stays the same, which means that it's actually cell dependent. But for most of it, it goes down. And then here's just another positive control called interferon alpha, and for all the cells, it goes down. The Zika production goes down, but again, we don't use that drug because it's harmful to pregnant women, and we don't want that to happen. And basically what this means is that if there's more soft phosphobutyric, then there's less Zika virus production, and then there's just less Zika, which is what we want. So here we see that it's dose dependent because, well, with 0, this is obviously without the drug, but then once taken 2 micromolar of the soft phosphobutyric, Zika replication goes down really quickly, and then more of it, when you give it more, it goes down even more. And that's because, like we talked about, eventually it'll run out, so the RNA polymerase will be able to add nucleotides that are the normal ones that it adds, but we want to keep the soft phosphobutyric there, so it'll add the soft phosphobutyric and then stop replication. So why were these past few pieces of data important? Does anybody want to tell me that? I'll see.
Well, it's pretty much proving that the drug did what it was supposed to do by decreasing the RNA polymerization.
Yeah, basically. So it just says that if there's more of the drug, then there's less Zika virus production / replication, which is what we said. So this is actually how it targets specific cell survival. So essentially this is important because it's targeting apoptosis of the cell, and this is just looking at it on a cellular level. And essentially this measures the y-axis measures the active caspases 3 and 7 over the total cells. And you can see that as the active cap space goes down, the more of the drug that's administered, the active caspase goes down, and the active caspases are enzymes that help in apoptosis. So if you have more of the drug, that means there's less apoptosis in your cell, in most of your cells, which means less cells are dying, and that's good because you don't want your cells to die. And then this one is kind of like the same thing, but essentially ETHD1 plus or positive, that's essentially it detects dead cells, and you can see that as soft phosphobutyric, the amount administered increases. The ETHD1 positive goes down, which means there's less dead cells. And the DPI is a stain that's used to get the DNA, and it does that through passing the cell membrane, and it can only actually pass through the cell membrane if the cell's dead. So as that also goes down, we can see that less cells are dying because less of those stains are going through the membrane. Yes?
Can you go back to the last slide?
Yes.
Did they establish a link? Did the paper clarify a link between Zika deregulation or whatever and cell survival? Or are they just two unrelated things?
I don't know, but I'd assume that they're two different things because, but the softball, like they're like kind of similar because they both like have the same goal of less replication with like less cells dying. So does anybody want to tell me what these two slides mean? Yeah.
It means that when you took the drug, you have less replication and less cell death. And that's kind of important because it shows that the drug didn't kill all replication, otherwise the cell would die from the specific only viral RNA.
Yes. So yeah, that's kind of what you said. And then right now this is like what they're doing right now. They're looking into why it doesn't affect the certain cells, like we saw before on the graph. 2 of the cells were straight lines, and those were like kidney cells. So they're looking into why that doesn't work. And they're also trying to create a vaccine because we talked about before how they have no vaccine. And they're also trying to create more antiviral drugs just in case Zika does have another outbreak. And then that's it. So, like, anyone have any questions? Yeah.
Do you know if this is tested in a human population?
I think it's like in like it's not in a human. It's tested like on just Zika virus infected cells.
Was it
It's
approved?
FDA approved, so that's what
Then yes, FDA approved. Sorry. Anyone have any other questions? Yeah.
So I assume sofosbuvir is not harmful to pregnant women like those other two.
Yes, that is correct.
Okay.
Do you know any of the potential downsides they talk about in the FDA approval process? Like outside of the paper itself?
Well, they didn't like really talk about much, but what they did is it's like cell dependent, as we talked about, like we saw before. Some it doesn't work in some cells, and that's kind of an issue because what if you have Zika virus in those cells, then they can just replicate without anything stopping it. So I guess that's a possible downside. Yeah.
What kind of cells were the types of cells that the drug doesn't work for?
They work like Vero cells, and they're like kidney cells. I don't know why it doesn't work in them. I don't know why it doesn't work in them. Any other questions? Yeah.
At what point is this drug administered to the pregnant women? Is it like just before they give birth, or
It's like once the Zika virus is like found in their system, they would take it because it's like a pill, and then once you take the pill, it should.
you would continue to take it throughout?
I assume so, but I'm not entirely sure. Yes.
So, I guess, so only 1 in 6 Zika virus cases, I think it was 1 in 6 or something, that display symptoms. So I guess detection of Zika would be a kind of a problem. Could patients take this if they know that they're going into Zika areas, or would the side effects be it's too negative to take it as kind of a preventive?
I don't think so because it's not really like a vaccine that you can take. It's more you get the virus and you like see that you have the virus and then you take it because it's an antiviral drug. So it never actually. I don't think you're supposed to take it before as a vaccine. Any other questions? All right.
So you said the drug like prevents apoptosis in cells, but if a cell is potentially cancerous, did it talk about how this drug could like allow cancer to be more probable?
Wait, sorry. Can you repeat the question?
Yeah. Will cancer be more probable because less cells are dying? So, because in cancer, the gene that basically prevents apoptosis, which is why tumors form. Does that make sense?
It's
like-
not preventing
apoptosis of all cells,
Yeah,
just like
just viruses.
infected cells.
Yeah.
Okay.
Because just to clarify, it does only target the Zika RNA
Yes.
polymerase.
Okay, thank you.
Oh,
I think
sorry.
also another point is that I'm pretty sure, if you can correct me if I'm wrong, but Zika virus leads to apoptosis of cells, and so by inhibiting the replication of the Zika virus, you're reducing apoptosis. The drug isn't directly affecting apoptosis in other cells.
Yeah. Any final questions? And then next week, Catherine is presenting.
Yes. So, just I guess
next year or this coming season, Matt and Kelly will be taking over very soon, so say hi to them. And Kelly's going to be telling you exactly what's going to be presented